Hello students, today we'll be continuing with the same chapter, that's the principles of inheritance and variation, and this is your second session. Today we'll be studying codominance, incomplete dominance, and the dihybrid cross, and the chromosomal theory of inheritance. So first let us study the incomplete dominance. So this difference between incomplete dominance. So incomplete means it's not completely dominant. It's a condition where the F1 has a phenotype that did not resemble either of the two parents. Means it's not completely dominant. This codominance means it's a condition where F1 generation resembles both parents. Word itself incomplete means not completed. It doesn't have resemble of two parents. Whereas codominance means in F1 generation itself it will be having the resemblance of both the parents. See, now these flowers, they look very beautiful, no? So these are snapdragon flowers and they come in different colors. Hybridization experiments were conducted on these pea plants. These were also conducted on snapdragon plants. So apart from snapdragon plants, the experiments were conducted on birds like pigeons and poultry, and also on several animals like mice, rabbits, cats, guinea pigs, and even man. So however, it was discovered that the law of dominance did not hold true in all the cases. So that is, Mendel's laws were not universal in occurrence. It was not accepted. So to understand this one better, now let us study the experiment which was conducted on the snapdragon plants. So when a true breeding or a pure line red flowered snapdragon with genotype capital R and capital R was crossed with true breeding white flowered snapdragon with genotype small r and small r. So the results, they were quite different. The one which was observed in pea plants were not seen in this. So the flowers in the F1 hybrid were pink and the genotype was capital letter R and small letter r. So that is, the F1 hybrid flowers had a phenotype that resembled neither of the parents but was in between the two. See, now the flowers of the F1 hybrid was taken and these were self-pollinated, and the genotypic ratios obtained in the F2 hybrid were 1:2:1. The same result was obtained by the Mendel in his experiments on peas. So however, the phenotypic ratios obtained in the snapdragon plant was also 1:2:1. So which is different from the ratio 3:1 dominant to recessive ratio in the pea plant. So in case of incomplete dominance, both genotypic and phenotypic ratios are same. That is 1:2:1. Thus, when a dominant allele does not completely mask the phenotypic expression of the recessive allele in a heterozygote, a blending of both dominant and recessive traits takes place in the F1 and F2 heterozygotes. So this phenomenon is called incomplete dominance. This is why the phenotypic ratios obtained in the experiments on the snapdragon plants were different from those in the pea plant. It is a gene for the flower color that shows the pigment in petals. Each allele is a code for the trait. That is, a specific amount of pigment. When both alleles of pigment are present, the petals are dark red due to heavy pigment production. On the other hand, if none of the alleles for the pigment exist, the flower is white. So, however, when only one of the alleles is present, only half of the pigment is produced, creating a pink shade. So, the alleles are therefore exhibiting incomplete dominance, which is a form of intermediate inheritance, where one allele for a specific trait is not completely dominant over the other allele. But why is it some alleles are dominant while some other recessive? Now we will have this doubt. No? So now let us understand this by studying the functioning of the gene. So you know that gene is a unit of the heredity that occupies a fixed position on a chromosome. That is called the locus. So a gene generally has two forms called alleles. In homozygotes, the alleles are the same. As in heterozygotes, they are not same, they are different. An allele may be different due to certain changes it has undergone, which modify the information contained by that particular allele. Now, example. Now, let's consider a gene that contains information for producing an enzyme. So, this gene contains two copies, which are the two allelic forms. Now, let's assume that the normal allele produces normal enzyme that is required to transform a substrate. Yes. So, according to some facts, the modified allele could be responsible for the producing a normal enzyme, a less efficient enzyme, a non-functional enzyme, or no enzyme at all. Now, if the modified allele produces a normal or a less efficient enzyme, we can say that the modified allele is equivalent to the unmodified or the normal allele. So, it means the modified allele will produce the same phenotype, or in other words, you can say that it can bring about the transformation in the substrate. Yes. Such equivalent allele pairs are quite common. Now, however, if this modified allele produces a non-functional enzyme or no enzyme at all, then the phenotype will be dependent on the functioning of the unmodified or the normal allele. So, here the unmodified functioning allele or the normal allele that represents the original phenotype, it is the dominant allele, and the modified allele is generally the recessive allele. So, now in this example, what we saw is that the recessive trait was due to the non-functional enzyme, or because no enzyme was produced. So, now therefore this incomplete dominance, it causes a distortion of the normal phenotypic ratio. It will disturb and will result in the offspring with the combined phenotype. So, this is a homozygous bull which is white in color with a genotype capital W and capital W, and this is a homozygous goat in color with genotype capital letter R and capital letter R. So, what color will be the calf be? It might. So, it is not white, or it's not red, or it's not a color in between also. So, the calf is wrong colored. Its coat is mix of red and white hair. So, therefore, in this case, The F1 generation, it doesn't resemble either of the parents, as in dominance, or it's not in between the incomplete dominance. But the calf displayed was the co-dominance. So co-dominance refers to a situation where a heterozygous organism has a phenotype that demonstrates traits from both dominant as well as recessive genes. So co-dominant trait is not blended, but it is independently and equally expressed. So another widely known example of the co-dominance in humans is the ABO blood system, which is used to determine human blood type. The plasma membrane of the red blood cells, or the RBCs, is present in the human blood as sugar polymers that protrude from its surface. So it is these sugar polymers that determines the specificity of the major blood types. For example, the difference between blood types A and B lie in a single sugar unit that protrudes from the end of a carbohydrate chain of a glycoprotein or glycolipid on the plasma membrane of an RBC. So it is a gene I that is responsible for determining the type of sugar produced by the RBCs. So this gene has three different alleles, namely capital IA, capital IB, and small I. So of these, capital IA and IB produce sugars that are slightly different from each other, while the allele small I doesn't produce any sugar. So here the alleles capital letter IA and capital IB are completely dominant over the allele small I. So now human beings are their diploid chromosomes, and hence they possess only two or the three alleles or the gene I. So consequently now we can see that there are 6 possible combinations of the alleles. Therefore, if the allele from one of the parents is capital letter IA and from the other the small I, only capital IA is expressed in the offspring, as the small I does not produce any sugar. So in this case, the RBCs only have type A sugar in them, and the genotype of the offspring is capital letter IA and small I, and the phenotype of the blood group is A. Similarly, when the allele from one of the parents is capital letter IB and from the other is small I, only capital IB is expressed in the offspring, as the RBCs have only type B sugar in them. In this case, the genotype is capital IB, small I, and the phenotype is B. So, however, when the allele from one parent is capital letter IA and from the other is capital letter IB, both the alleles expresses their own types of sugar in the offspring due to co-dominance. So, in this case, the RBCs have both A and B types of sugars in them, and the genotype of the offspring is capital IA and capital IB, and the phenotype of the blood group is AB. Now, if the alleles are from both the parents are capital letter IA, once again the RBCs have only type A sugar in them, and therefore the offspring has the genotype capital letter IA, capital IA, and phenotype A. In the same way, if the alleles from both the parents are capital I and B, and they're having only type B sugar, and so the offspring will be having the genotype capital IB and capital IB and phenotype B. However, now if the alleles from both the parents are small I, the RBCs will be having no sugar in them, and the genotype of the offspring is small I, and the phenotype is O. Therefore, as there are 3 different types of alleles, there are 6 possible genotypes and 4 different blood groups or phenotypes that exist. Apart from being good example for codominance, the ABU blood grouping system is also an ideal example of multiple alleles. So now we saw that the gene I has 3 alleles, capital IA, capital IB, and small I, that govern the same character, that is blood group. So however, since an individual can have only 2 alleles, it is possible to learn about multiple alleles only when detailed population studies are conducted. So according to the multiple alleles concept, more than 2 alleles govern the same character. Now let us study the experiment which shows that single gene may sometimes produce more than one effect. These seeds are having a single gene with 2 alleles, capital B and small b, that controls starch synthesis in them. So in the experiment conducted, the effective starch synthesis was seen in capital B homozygotes, and the seeds produced were large amount of starch in them. Moreover, the pea seeds were round. On the other hand, the small b homozygotes were not very efficient at synthesizing starch, and so the pea seeds produced were small or had less starch in them. Also, the seeds were wrinkled. Moreover, in the experiment conducted, the seeds or the starch grains produced by the heterozygotes, capital B and small b, one of the intermediate size, but they were round. So in this experiment, if we consider the starch size as a phenotype, then the alleles displaying incomplete dominance. On the other hand, if we consider the shape of the seed as a phenotype, then the alleles are displaying the dominant as the seeds were round like one of their parents. So now this experiment shows that the dominance is not an autonomous feature of a gene or gene product. Therefore, dominance, incomplete dominance or codominance depends on the gene product and the production of the particular phenotype from this product. And it also depends on the phenotype that we finish to examine in the case the same gene influences more than one phenotype. So Gregor Mendel also cultivated and tested about 29,000 pea plants between 1856 and 1863. So one such test was the crossing of the pea plant with yellow and round seeds and those with green and wrinkled seeds. So as you know, such a cross between plants that differ in 2 traits or characters is known as dihybrid cross. So this experiment conducted by Mendel resulted in a pea plant that produced yellow and round seeds in the F1 generation. Here, now can you guess the dominant shape and color? Yes, it is round shape is dominant and the wrinkle shape, and the yellow color is dominant over the green. Mendel noticed that these results were identical to the results he had obtained when he had carried out monohybrid crosses between the plants with round and wrinkled seeds and those with yellow and green seeds. Now in Mendel's dihybrid experiment, let's denote the dominant yellow seed color with the genotypic symbol capital Y and the recessive green seed color with small y. Likewise, now let's denote the round seed as the capital letter R and the wrinkled seed as small r. The genotype of the parents can therefore be denoted as capital R and capital I and capital Y, and small r and small y. So on crossing the plants, the gametes capital R and capital Y and small y unite to produce yellow and round seeds with genotype capital R and small r, capital Y and small y in the F1 generation. Mendel further self-pollinated the plants obtained in the F1 generation to produce F2 generation plants. So this time he found that 3/4 of the seeds were yellow, 1/4 was green. So similarly, he noticed that 3/4 of the seeds were round while 1/4 was wrinkled. Therefore, the yellow and green color had segregated in the ratio 3:1, similar to the results obtained in a monohybrid cross, and the round and wrinkled shape had segregated in the same ratio 3:1. Mendel he also found the phenotypic ratio in the F2 hybrid was 9:3:3:1. That is, there were 9 round yellow seeds, 3 round green seeds, 3 wrinkled yellow seeds, and 1 wrinkled green seeds. Mendel had got the same phenotypic ratio in several dihybrid experiments that he had conducted. This ratio 9:3:3:1 can be derived as the combination series of 3 yellow:1 green with 3 round:1 wrinkled, and further this derivation can be written as shown. So from such observations, Mendel proposed a new set of generalizations, which is later called the law of independent assortment. So this states that when two pairs of traits or characters are united in a hybridization experiment, the segregation of one pair of characters is independent of the other pairs of the characters. So now we can use the Punnett square to easily understand the independent segregation of two genes during meiosis and the production of pollen grains and eggs in the F1 generation. So now for example, in the dihybrid cross experiment which is conducted by Mendel, the genotype of the seed obtained in F1 generation was capital R, small r, capital Y and small y. So in the segregation of one pair of genes, capital R and small r, here are 50% of the gametes, and they are having the capital R gene, and the remaining 50% are having the small r gene. So other than this, gametes having capital letter R, small r, and it must also have either capital Y or small y. So here it is important to note that the segregation of 50% capital R and R, 50% small r is different from the segregation of 50% capital Y and 50% small y. Hence 50% of the gametes bearing small r has capital Y and the remaining 50% has small y. Similarly, 50% of the gametes bearing capital R has capital Y and the remaining 50% has small y. Therefore, the genotypes of the gametes are not of 4 types, namely capital R, capital Y, capital R and small r and capital Y. and small r and small y. So in other words, there are 4 types of eggs and 4 types of pollens. And you can see that the frequency of these gametes is 25% or 1/4 of the total number of gametes produced. So now the allelic composition of the zygotes of the F2 plants can be easily obtained using the Punnett square as shown. So as you already saw, there are 4 different types of phenotypes found, and phenotypic ratio is 9:3:1. So however 9 different genotypes results from F2 hybrid, and genotypic ratio is 1:2:2:1 4:2 and is to 1 and is to 2 is to 1. So thus from the dihybrid cross experiment, Mendel found that the gene combination of progeny in the first are the parental gene generation combination. The gene combination, Gregor Mendel, he is the father of modern genetics. He has not only done research in pea plants but also on the several experiments like honeybees. So in fact, by 1865, Mendel had published his work on inheritance of characters. However, his work did not receive any recognition until the beginning of the 20th century due to several reasons. So firstly, Mendel's work could not be widely published because means of communication in those days were not well developed as they are today. Secondly, Mendel had considered genes of factors as discrete and stable units responsible for controlling the expression of traits. He had also explained the concept of alleles and how they were responsible for the continuous variation observed in nature. So, however, his concept about genes were not readily accepted by his colleagues. So, thirdly, Mendel, for the first time, had applied some statistical analysis and mathematical logic to the problems in the biology, which was objected to by many biologists because he was a mathematician, no? So they were not ready to accept it. Finally, Mendel, he could neither explain what genes were made or provide any physical proof for their existence. So Mendel's work got credits only during 1900 on which were independently rediscovered by the 3 scientists Hugo de Vries, Carl Carnes, and Eric von Schermach. So also both the chromosomes and the genes segregate during the process of gamete formation, and only each of the pair is passed to a gamete. However, they differed in the way they segregate. So in genes, independent pairs segregate independently of each other, while in chromosomes, one pair segregates independently of another pair. So now let us study the behavior of the chromosomes during cell division. So in the anaphase stage of the meiosis 1, a set of chromosomes aligns along with the metaphase plate. Here now we can see the long orange and a short green chromosome, as well as long yellow and short red chromosomes, which is lying at the same pole. So now you consider another set of chromosomes aligned along the metaphase plate during the anaphase stage of the meiosis. So here now there is a long orange and a short red chromosome, and a long yellow and a short green chromosome at the same bone. Now during the anaphase stage of meiosis 2, we can see that orange and green chromosomes segregating together in the cell on the left. While the orange and the red chromosomes segregate together in the cell on the right. So certainly he analyzed this segregation of chromosomes during meiosis and concluded that chromosomes are having individuality, and chromosomes occur in pairs with members of each pair contributed by each parent. So he also concluded that the paired chromosomes separate from each other during meiosis, and the distribution of the paternal and maternal chromosomes in each homologous pair is independent of each other. So both Sutton and Vavary were of the opinion that the pairing and separation of pair of chromosomes would result in the segregation of the pair of genes or factors carried by them. So their hypothesis on chromosomal behavior became to be known as the Vavary chromosome theory. So Sutton combined his hypothesis of chromosomal segregation and Mendelian principles, called it the chromosomal theory of inheritance. So students, these are the important questions. Define incomplete dominance. Explain incomplete dominance in Snapdragon. Second one, explain dihybrid cross with schematic representation using Punnett square. So these are all 5 marks questions. And third one, what is codominance? Fourth one, write the genotypes of blood groups in man. You have to write the blood groups and the genotypes. And fifth one, who proposed the chromosomal theory of inheritance? It's a 1 mark question. So who proposed this? It was proposed by Walter Sutton and Theodore Boveri. Any doubts you can ask me. Actually, it's very important chapter. If you don't understand it, please come out with your doubts. Thank you.